Pfaffenhofen in Upper Bavaria is probably one of the most attractive places in the world to live in. It is also an important industrial location. One of Daiichi Sankyo's European production sites is located there as well. Pharmaceuticals are not only manufactured but also developed here. The goal of pharmaceutical development is to produce applicable medicines such as tablets from active ingredients. The product formulation and analytical departments work together closely in order to achieve this. The product formulation department identifies active ingredients and excipients of a medicine, such as fillers and binders, disintegrants, anti-adherents and coatings, and defines their mixing ratio. In order for an agent to be absorbed and metabolized well by the organism, the ideal particle size and the corresponding fineness have to be determined first. Once the right formulation is found, it's tested on the laboratory scale as a next step. This means the tablets are manufactured following the same steps and processes as they will be later in standard production, only on a much smaller scale. During the entire development process, previously defined work safety measures are adhered to, tested, and developed further. The work safety measures required depend on the active ingredient. The selected raw materials are subsequently mixed on the laboratory scale, then processed into granulate in a fluidized bed installation. The next step is to press the granulate to form tablets. In addition, coating is applied to the tablets. This helps to disguise the often bitter taste of the active ingredients, provides for improved abrasion resistance, and creates a predefined look. The prototypes manufactured this way are then tested in the lab. During dissolution testing, liquid warmed to 37 degrees centigrade is used to simulate how the tablets dissolve in the stomach. For this, the liquid is being moved by standardized paddles. This step in the development process is to test how quickly the active ingredient is released. Using the data from this test, the formulation of the pharmaceutical product can be improved further to achieve the planned effectiveness. Once the ideal formulation has been defined, all steps of the future production processes are tested and validated in a 30 kilogram scale. This is equivalent to about 10% of the later volume in a single production step. A near-infrared spectrometer is used to check, amongst other things, whether all raw materials are distributed evenly during the mixing process. During every step of production, like compression in this example, consecutive samples are taken for continuous testing. This is the so-called in-process control. It's used to test the weight, crushing resistance, abrasion resistance, and height of the tablets, and to measure the dissolution properties. This ensures that the millions of produced tablets all have the same properties. Part of every pharmaceutical product is suitable packaging. Particular attention is given to primary packaging, namely the packaging that's in direct contact with the pharmaceutical product. In the case of tablets, this comprises the cover foil of the blister packaging, amongst others. The composition of the materials is checked once again with the help of an infrared spectrometer. The foil's thickness is tested as well. For every new active ingredient and its dosage form, individually tailored methods are developed to investigate the product's quality and shelf life. With the help of innovative software solutions, the analytical department develops and optimizes chromatographic separation methods efficiently. These methods are particularly important for defining a new product's shelf life. In order to check how long a pharmaceutical retains its quality, stability samples are packed separately and stored for years in special climate chambers with precisely defined temperature and air humidity. Whether and how the tablet changes during this time frame is checked at regular intervals. The active ingredient and all byproducts are quantified during the process as well. If the employees find new degradation products, mass spectrometry is used to identify these products. All generated data and gathered information about the quality and shelf life of a new pharmaceutical product are an important element for regulatory approval worldwide. Before approval is given, the pharmaceuticals have to be tested in clinical trials, often with up to thousands of patients. The samples required for these trials are provided and sent around the world from Pfaffenhofen. Only once a trial has proven the effectiveness and safety of a pharmaceutical product will the required approval be given. Large-scale production and distribution follow the approval. Developing formulation and production processes for a new pharmaceutical product and then testing its stability takes 3 to 4 years in all. This time-consuming and diligent approach is a vital prerequisite to making high-quality and safe pharmaceutical products available to patients worldwide. Daiichi Sankyo, passion for innovation, compassion for patients.